Good day, this is Professor Will, CBMD PhD, and today is Tuesday, June 8, 2021. It is 1:27 and 5 seconds p.m. Eastern Standard Time. Today's research, biomolecular labeling, and feel free to check out my website. Well, let's hop to it here. Clever biomolecular labeling enables identification of immune cells. Research team led by the University of Göttingen develops new strategy for labeling peptides. Date June 7, 2021. Source University of Göttingen. Biomolecules regulate the biological functions inside every living cell. If scientists can understand the molecular mechanisms, then it is possible to detect severe dysfunction. At a molecular level, this can be achieved with fluorescent markers that are incorporated into the respective biomolecules. Researchers are now able to show that a complex manganese makes it possible to conveniently label certain biomolecules. In the past, this has been achieved by incorporating a marker in the biomolecule by completely rebuilding it from the beginning, necessitating a large number of steps. Unfortunately, this approach not only takes a lot of time and resources, but also produces unwanted waste products. Researchers at the University of Göttingen and Edinburgh have now been able to show the non-toxic complex of the common metal manganese makes it possible to conveniently label a class of special biomolecules known as peptides right at the last minute of their synthesis. This means the mechanism of action of these labeled peptides can be investigated efficiently. The research group developed the selective labeling of peptides and natural biological products at a late point in the series of steps necessary for synthesis by activating carbon-hydrogen bonds in tryptophan residues. This experimentally simple strategy makes it possible to efficiently access novel fluorescent peptides that are highly sensitive to their biological environment. This enabled the team to create a highly sensitive rotor with the ability to show changes in the composition of membranes of immune cells. Its fluorescence is dependent on the viscosity of the cell membrane. The researchers observed radiant fluorescence in the presence of cholesterol in the cell membrane. In this way, the rotor can be used to screen certain molecules in cells that are important for the adaptive immune system to fight infections and cancer. The project demonstrates the power of combining chemical, biological, and medical research, which allows a direct observation of cell-specific events. Furthermore, the successful collaboration ensures our discoveries to have an immediate impact not only in the field of chemistry, but also in the biomedical sciences. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video is created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.